Okay, so let's get started for this week. So, just a recap about what we studied last week. So, we studied constraint-based modeling. One of the types of constraint-based modeling was FPA, balance analysis. We studied that. We studied how to do based analysis, flux balance analysis, and the other versions of flux balance analysis like Room or MOMA. Today we are going to look a little bit more into constraint-based modeling. So another variety of flux balance analysis. We'll look into flux variability analysis and some important properties of metabolic models and how do you tackle dead end metabolites and other uh, other stuff in metabolic models. So let's get into it. Okay. So first topic that we'll cover today is the gene protein reaction associations. So this is a schematic given trying to explain what gene protein reaction associations are. So just a second. Okay. So let's get started. So first we start with the concept of gene protein reaction association. So this is a schematic that describes different gene protein reaction associations. Sorry. Yeah. So the first panel on the left is shown as a simple association in which a single gene encodes a single enzyme. So a single gene encodes a protein. The protein here is the enzyme which takes part in the reaction. Now there can be other varieties of these gene protein reaction associations. So the second panel that you see, these are what are known as isozymes. So multiple genes, here gene 1 and gene 2, encode distinct proteins, but they carry out the same function. So gene 1 and gene 2 are producing two different enzymes which are basically similar in function, can catalyze similar type of reactions. So if you block one of the enzymes, the other enzyme will take its place. So these are known as isozymes. So these are distinct proteins, different proteins that are produced by different genes, but they have similar functions. Uh, Another variety of these gene protein reaction associations is in this panel number 3. These are known as multimeric protein complex, where multiple genes encode distinct protein subunits that come together and form an active enzyme. So, enzymes, a little bit of biochemistry refresher. So, basically, enzymes have different domains and different units. So, there is the enzyme binds to the substrate at a place known as its active site. So there are other sites like active sites inside the on the enzyme which have different functions. So there can be other sites which go and help bind the enzyme to the protein, or there are sites that help the enzyme bind with each other to form dimeric enzymes and stuff like that. So and these enzymes can come together, different subunits of an enzyme can come together to form the full functional unit. So this is what the multimeric protein complex is depicting. So you have different genes encoding from different subunits of the enzyme, which basically is accumulated or taken together, all the subunits, they form like the functional enzyme. So uh, this is basically multimeric protein complex. Now, another variety you have is basically multifunctional protein, which a single gene is encoding a single protein or a single enzyme, but that protein or enzyme is involved in multiple of reactions inside the cell. So there are maximum of the enzymes that you see in the cell are of these type, multifunctional types, because like in each metabolic pathway, no, basically a single enzyme can be shared across different metabolic pathways or different, in general, signaling pathways in the cell. So there is no like rarely you'll find a one-to-one mapping between an enzyme and a particular metabolic pathway, because that enzyme may be involved in other metabolic pathways as well. So it's kind of a one-to-many mapping that you find. Okay. Moving on. So now we'll discuss what are metabolic reaction deletions. So basically, we studied how in the last class using flux balance analysis, how you can model this metabolic pathways and networks and get an understanding about how the flux is flowing and what kind of growth rates you can have from the network and etc. etc. This is one of the techniques where you try to perturb the metabolic network or the metabolic pathway. So basically, you're trying to delete a metabolic reaction. There are various ways to do it. One of them is depicted here. So metabolic flux balance analysis is a computational method that uses genome sequencing and biochemical databases to study organisms' metabolic capabilities. We studied how FBA works in the last class. So yeah, FBA can be used to simulate the effects of in silico gene deletions. Now, as I was saying, the relationship between genes, proteins, and metabolic reactions is not often not one-to-one. So as I was saying, a single uh, metabolite or a single enzyme can be involved in different multiple reactions. So these enzymes catalyze these reactions, but multiple enzymes can catalyze the same reaction, and one enzyme can catalyze multiple reactions. So these multiple enzymes catalyzing same reaction is the isozyme example, this example, and a single enzyme catalyzing multiple reactions is this fourth example, which is the multifunctional protein example. So yeah, now, deleting a reaction may require deleting multiple genes because multiple enzymes and multiple metabolites are involved in that reaction, which are get encoded by different genes altogether. So, the removing a reaction may require deleting all the genes that are producing that particular substrate or particular enzymes. Now, yeah, basically this can alter the metabolic solution space. For example, removing a specific reaction may improve production rates, but deleting the genes associated with the reaction can lead to a lethal growth phenotype as other essential reactions may be affected. Additionally, if a reaction lacks known associated genes, genetic engineering may not be feasible. We'll talk about a little bit about the difference between gene deletions and metabolic reaction deletions. Sir has also covered that in the video, but yeah, we'll talk about that a little bit in the next slide. So this is a schematic showing the modeling framework that we have for metabolic models. So this is a metabolic network or a model that is represented by the combination of three components. So you have GPR Boolean rules associated genes, G1 to G5, five genes associated. You have three reactions associated with these five genes. So these are the Boolean connections between these genes, like G1 or G2 or G3 can like form a enzyme that can mediate this reaction. G3 or G4 can form an enzyme which can mediate the R2 to R3 reaction. So these are Boolean rules that are associated with the model, and uh, basically you have three reactions as well. The network of reactions defines a set of feasible biochemical transformations. So basically, uh, I think these are the biochemical. Basically, the network of the reaction defines the set of feasible biochemical transformations, and a set of biomass precursors defines the requirements for growth. So biomass, there are certain biomass precursors which will account for your in your model that will help that are basically required for growth. And growth phenotypes are predicted by assessing whether all biomass precursors are produced by the metabolic network from the set of metabolites in the reaction. uh, Gene deletions potentially can inactivate reactions, which in turn may reduce the space of producible metabolites. So you see, like previous possible basically biomass space, basically the biomass um, basically this is your metabolite feasible solution space, which is basically coming down here after you delete this gene. So if you delete this gene, this is essential for this R3 reaction, which is basically inactivated due to the deletion of this G5 gene, which basically in turns reduces the producible metabolite space. So this space is further reduced. So there's one precursor that is no more producible. So you had certain precursors which were required for growth. Now one precursor of this, basically one solution of this, is falling out of the metabolite space after you delete a gene. So this is one of the examples of how you can basically how you can handle gene deletions in a network. But there are other methods as well. We'll not go into details of all those, but basically the idea is you need to account for which genes are getting deleted and how each how the reaction set of your metabolic model are getting affected because of the deletion of the genes. So these are the two main important things to keep in mind. Now, this is an example. This is basically showing the differences between reaction deletions and gene deletions. So, this is an example, basically gene to protein reaction association for these enzymes TKT1 and TKT2 in E. coli. So, you have this. These are two genes which encode for TKTA and TKTB, which basically like TKTB and TKTA are both involved in the reaction of producing TKT1. Same is the case of TKT2. Now, the solution space, basically the wild type is shown by the blue and the mutant is shown by the green. So the solution space for wild type blue and TKT1 PTAR reaction deletion strain, which is shown by green, predicted by the metabolic model. So this is your wild type basically solution space, and the green is your mutant solution space. Just a second. Yeah, so your blue is your wild type solution space, and the green is shown to be your mutant solution space. Now you do certain gene deletions. You delete TKTA, B, PTA, and EUTD. So basically, these are corresponding genes that are associated with the reaction of forming TKT1 and TKT2. Now, when you delete all these genes and all these genes associated with this reaction to eliminate basically TKT1 or PTAR, no growth is predicted for the mutant strain. As you can see, the blue one is showing your wild type uh, solution space, but your mutant solution space is shown by this line, which is basically parallel to the y-axis. If you can see, there's a bold line here. So basically, your solution space is only deciding on the y-axis. So basically, you have no growth. So if you delete reactions, as you can see, if you delete certain reactions, you can still get a growth of the mutant. Your model may still predict growth of the mutant, but when you delete these genes that are associated with the reaction, your model is predicting no growth. Now, biologically speaking, there can be various reasons for this. So first, and the most important reason is basically these genes are not only associated with this particular reaction, they're associated with in the overall survival of the cell. So basically, if you delete these genes, you're not only affecting the current reaction or the set of reactions, you're affecting the overall survival, because these genes are essential in basically helping the cell survive. Now, uh, biologically thinking, this is a very logical explanation to the point that your model is predicting that you have no growth in in basically your metabolic model. So reaction deletions and gene deletions are something that are clearly very distinct things, and we need to know the differences between those. And when we apply either of them, we need to know what kind of repercussions we might see. So reaction deletions, there's a much greater probability that your system will still survive with a certain level of growth, maybe less than the wild type space. So for in this example only, like the wild type space is a little bit bigger than the mutant space. But when you delete these genes, it is very much possible that your cell will not survive. So you have to be a little bit careful about it. So yeah, this is what I wanted to discuss: reaction deletions versus gene deletions. Okay. Next, we move into the concept of dead end metabolite. So a dead end metabolite, what it is basically, if you define a dead end metabolite, it is a metabolite that is produced by the known metabolic reactions of an organism and has no reactions consuming it, or that is consumed by metabolic reactions of an organism and has no known reactions producing it. And it both cases has no identified transporters. So basically, if you see in this panel, this is a representation of showing dead end metabolites. So uh, you have four components here. You have A, you have B, you have C, and D. The compounds A and C are neither produced nor transported by any other reaction within the network. So A and C are not produced or transported by basically by any kind of reactions or any kind of transporters inside the cell. So your A is not produced here. A is just, if you imagine A to be an ATP. So if in your reaction ATP is getting used, but it is ATP is not formed in this reaction. So this A is a compound that is getting used in the reaction, but which is not formed, and A is not transported or to by any other reaction within the network as well. Similar is a concept for C. C is getting converted to D, but there is no the C is not produced, neither produced inside the metabolic environment that you are looking at, and also C is not transported by any other reaction. So these are basically neither consumed nor transported by any other reaction in the network, and mm, which essentially means that these are isolated compounds within the metabolic. So a dead end metabolite are basically those metabolites which are neither produced nor consumed inside the network, uh, inside the metabolic space you are looking at, and these are isolated compounds within the network. In some cases, these dead end metabolites reflect a deficit or an error in how a metabolic database represents knowledge from the scientific literature, and alerts us to the need for further curation of the database. So it can happen that you have missing information from the database that you are getting your data from. It can happen that your some information is missing, which is leading to the fact that some of your metabolites in the metabolic space you're looking at are basically dead end metabolites. That can happen. That is like, if that happens, that's not necessarily a issue of your model. That's an issue of missing information. But in some other cases also, this systems level analysis alerts us to areas which are more experimental research, in where more basically experimental research is required. So basically, if you're looking at a metabolic network and you find certain dead end metabolites, it is possible that no data is available from uh, on those particular metabolites from scientific literature, because no experimental research has been done on that only. So it is very much possible to identify those dead end metabolites uh, after like curation from all the scientific literature. You can identify certain dead end metabolites in your model, and which needs basically further information for your model to be completed. So those information you will not get from your model. You need experimental research to do so. So you can like show the need for doing experimental research using dead end metabolites, because these are generally compounds that we do not have information on. So yeah, these are what dead end metabolites are. Next, we come to the concept of block reactions. So initial metabolic network reconstructions often contain inconsistencies due to annotation errors or unknown enzyme functions. These inconsistencies lead to missing reactions in the draft model, creating gaps in the metabolic pathways. So, like, there are certain as I'll explain it after I finish this slide. So, dead end metabolites, which are classified as root non-produced or RNP, or root non-consumed or RNC. So here I talked about the dead end metabolites. They are neither produced nor transported by any other reaction, or and is neither consumed inside the reaction. So if it is not consumed, we uh, called it basically root not consumed, and if it is not produced inside the network, we call it root non-produced. So we have RNP and RNC. Basically, it's how the name suggests. So RNP are metabolites that are only consumed by the system's reaction, and RNC are those metabolites that are only produced by the network but are never consumed. So RNP RNC block reactions causes further gaps downstream or upstream of metabolites. So if you have like basically dead end metabolites, so it could, what it contributes to the model is basically incomplete reactions. So these are gaps in your model because you do not have the information, or you do not have the whole reaction information available to you. So identifying these inconsistencies, these dead end metabolites and these blocked reactions is known as the gap finding problem. So what are gap metabolites? This is a schematic showing four classes of gap metabolites that are basically consequence of missing reactions. So this red cross indicates the absence of some reaction because you do not know from where from this A is coming. So that information uh, is not available, which classifies into the group of RNP, root non-produced, which is basically A is not produced inside the inside your metabolic space, but you do not know basically from where A is coming. So yeah, this red crosses on the both side. Here basically it's an RNC. H, it is neither transported nor consumed, so it is root non-consumed. But yeah, so these two red crosses are depicting RNP and RNC. So these indicate some sort of absence of some reaction, and these dotted and continuous arrows represent blocked and non-blocked reactions. So the continuous arrows are basically non-blocked reactions. The dotted arrows are showing blocked reactions. And these yellow and green circles represent gap and non-gap metabolites. So green are the non-gap metabolites, E and B, and rest of them, A, B, C, and F, G, H, are basically gap metabolites because you do not have enough information uh, on them. And basically metabolites are labeled according to its class. In the absence of a reaction causes metabolite A to become RNP, as I discussed. So you do not know from where A is coming. It's not produced inside the space, so it's RNP. And this affects basically, since you do not know from where A is coming, you do not know the stoichiometry of A. So this, what it does is it affects the effect of this propagates downstream, creating new gap metabolites. So these are known as downstream non-produced DNP and blocked reactions. And because you do not know A, the stoichiometry of A, you do not know the stoichiometry of B or C. But on D, because D is a production pathway, you know how much of D is producing how much of your growth or how much of your particular metabolite is coming out. So the end of the reaction, you know, okay, like what is happening. But before that, prior to that, you do not know how much of A is giving how much of B or how much of C. That information is unavailable. Similar in this case, the the consumption and information is available, but the production information it's not. So basically, you know how E is getting consumed, but you do not know how much of H is getting formed, or like basically how is E forming F, G, and H, and how H is getting ultimately consumed. So it the caption also says the same. The absence of reactions consuming H makes it a RNC, and this effect propagates upstream, causing other metabolites to become upstream non-consumed, basically UNC, in a symmetrical manner in respect to case A. So the take home message from this slide is basically, if you have certain gaps in your model, so you have certain basically you have certain dead end metabolites. So these metabolites are not only affecting your basically the particular production of that particular metabolite, it is also affecting downstream and upstream metabolites as well. So these are the things that you need to keep in mind when you're making this model. So there can be blocked reactions, there can be gaps, there can be dead end metabolites, which you have to, like, these are basically information gaps inside your metabolic model, and you need to consider all of this while trying to model the metabolic reactions. Okay, so next we come to the concept of what is known as flux variability analysis. So we studied flux balance analysis in the last class. So this is basically a different version of basically flux balance analysis, where you have certain variability in your solution space. So in a flux balance analysis, you projected the solutions on your solution space, and you had a certain section of the solution space which is giving you a set of solutions. Now these solutions that you are getting from FPA were very rigid. They were like not basically they were particular values. Now there is some variability associated with those results as well. So that is what FPA in principle does. So flux balance analysis predicts steady state of fluxes of reactions in a metabolic network based on biological imperative. So according to how you model, how many constraints you put and everything, FBA will predict steady state fluxes of the reaction. So SV equal to zero. I hope you remember that. So that's the form that you have, and at steady state SV equal to zero. From there, FBA will give you steady state fluxes of these reactions. Now, FBA will not always yield a unique solution, prompting the need of flux variability analysis. So you can, it's very possible that FBA will not yield a unique solution. Now, what flux variability analysis does is it quantifies the feasible ranges of reaction fluxes at optimal or suboptimal production rates determined by the FBA. So it is basically a generalization of FPA, and it ultimately aids in understanding the metabolic network flexibility. So applications of FPA are various. So I've listed a few down here: medicine, health, etc., etc. And it enables analysis of reaction flexibility within metabolic networks, offering insights into system behavior. Now, this is one of the most important points of FPA, that it enables analysis of reaction flexibility. So this variability that you're getting with the particular fluxes. So there, you can have a maximum and a minimum value. So you have always a plus minus your whatever be it, standard deviation or standard error or whatever your is the variability factor. So there is a maximum of this variability and there is a minimum. So you have a range of solutions for a particular metabolite, which gives you basically a reaction flexibility within the networks. So basically, your reaction can have the maximum flux, which is corresponding to this particular value. Your reaction can also have a minimum flux corresponding to this particular value. Now, your reaction can be within this range. Now, within this range of different fluxes of this, basically your variability parameter, there can be different solutions of these metabolic networks. So basically, if you see a particular reaction is when it is at its highest, the other reaction is at its lowest. So it gives you an idea that maybe this reaction, like speeding of speeding up of particular reaction, is basically slowing down the other reaction. So this basically flexibility in terms of the solution space, this is what FPA primarily gives you, and it can give you insights into systems behavior. Like I said, like if a particular reaction having the highest basically you have a range, you have a max and mean. So particular reaction uh, occurring at the maximum value is pushing another reaction to perform at its minimum value, is showing some sort of a negative correlation or something of this sort. So systems level uh, interaction and behavior can also be modeled using FPA. So this is the general overview of FPA. So first you identify the maximum value of the objective function and constrain the objective function to this value. And second, you minimize and maximize each flux independently to identify flexibility in fluxes across alternate optima. So this is basically the second point is what I just mentioned. So you have a maximum and a minimum value for each flux, each metabolite involved, and you can basically leverage this flexibility and find different fluxes across alternate optima. So like you can find different fluxes for different metabolites under different conditions. So you can even make predictions like this particular condition, this is how the metabolic network works. Under this particular condition, this is how the metabolic network works. This is the range of fluxes that you can have for this particular metabolite according to the given constraints. And within that range, you can have different types of solutions which can basically upregulate or downregulate other reactions as well. So this is what you have as an overview of FVA. So this is just an information uh, that you might like. So basically, there's a mathematical proof involved, but in general, if you have n fluxes, you need to solve two n FPA problems according to if you are doing flux variability analysis. So this is just a general statement. It's sometimes more, sometimes less, but it's almost two n. So yeah. So this is a schematic which describes the FVA. So you determine what is possible based on external measurements and network stoichiometry. So you have I metabolites and J reactions, and you have a feasible region. So you have basically you have a maximum and a minimum function of this VJ. So basically you are putting constraints on your solution space, and this is your basically a steady state function sp equal to 0, and you are putting a constraint there. Your vj lies between vj minimum and maximum. So this maximum and minimum is given by the variability. It's given by the standard deviation on the standard area, that plus minus that you write. It is this minimum and maximum is given by that, and this is your basically c dot v is your optimizing function, or your basically cost function. So for each reaction J, you need to find this basically this feasible region, and you try to characterize this feasible region. Okay, like what may be happening or how it may be affecting other reactions as well. Now, if we huh. So this is one example that I have put in for FVA. So I'll go through the example and how it works out. So basically, if FBA doesn't always yield a unique solution, alternate pathways can achieve the same objective, resulting in phenotypically different solutions. Now, FBA identifies maximum and minimum fluxes through reactions while maintaining optimal objective values. So this is what we discussed in the previous slides. Now, uh, how you can do FPA. So there is this Cobra toolbox. I hope you have seen the videos for it in the course, but I'll not go into the detail of the Cobra toolbox. I'm just giving an example of like how basically in real world how this flux variability analysis is done. So you have two flux maps for two alternate solutions. These are basically solutions. I'll come to the solutions later. So this is how your solutions look like in general. I'm just giving you an overview. So these are two flux maps for two alternate solutions of for maximum aerobic growth on succinate. So in your metabolic model, you basically this involves metabolic pathways. Like this is I think is a Krebs cycle. There is glycolysis as well involved. There are other metabolic pathways involved as well. So what you want to do is you want to maximize your production of succinate, your aerobic growth on succinate, and like as we saw that FBA always doesn't give that unique solution. So these are two alternate solutions from basically FBA. Uh, how are they different? In the first one, the reaction Me1 is used to convert N-malate to pyruvate, but in the second part, this reaction is not used at all, and the reaction P/K is used. And these two alternative reactions are highlighted in red. So this is one alternative, and the other alternative is this basically. So uh, yeah, so these are two different solutions for the same optimizing problem. Now, how do you do the FBA on this? So you have your Cobra toolbox. So there are different functions defined inside this Cobra toolbox. So you can change your reaction bounds using this. You can set your objective function, your optimizing model, everything for the FBA. Now, after you have all the toolboxes, you obtain an optimal objective value for your cost function or your Z, and you set upper and lower bounds of this objective reaction to Z. So you have an objective value, you set an upper and lower bound, and you set the reaction of interest as objective. So your objective function should try to maximize your, in this case, your aerobic growth on succinate. In other cases, depending on your model, what you're trying to maximize. And then you use FPA to minimize and maximize this new objective separately. These will yield maximum and minimum possible fluxes throughout the reaction while maintaining that optimal objective value. Now, this is the details of their example. So you have the Malik enzyme reaction ME1 in E. coli growing on succinate. So the minimum flux through it is basically 0, and the maximum is 6.49. And the two alternate optimal solutions are basically in which one Me1 is used and the other one in which Me1 is not basically used. So in this one, Me1 is used to convert this L-malate to pyruvate. And the second example, the Me2 is not used. Instead of Me2, there is another PYK reaction that is used. Now, uh, yeah, these are your conditions. So your minimum and maximum are 0 and 6.49, and you have two alternate optimal solutions. Now, how do you do FVA? So in your Cobra toolbox, it has that FVA function for on every reaction of the model. So this example is FVA on E. coli core model. So these models are available within Cobra toolbox. If you look, you'll get these. So this is an FAA on E. coli core model for succinate growth, identifies 8 variable reactions. So these are the reactions that you see in this table. These are the 8 variable reactions that you have from FV. Now, these variable reactions indicate different pathways for achieving maximum growth rate leading to alternate solution. Now, one thing to keep in mind, basically all your solutions are trying to maximize or minimize your Z function, basically your cost function or your optimizing function. So in this case, your optimizing function should be the maximum growth of on succinate. Now, both of these methods will give you maximum growth on succinate using Me1 or not using Me1, using PYK. So you have these two alternate solutions, but your ultimate goal is maintained as the same. So your ultimate goal is achieving maximum growth rate. Both of these solutions are giving you maximum growth rate. So these basically, like, these are the two alternate solutions that you can get from this FPA function. This is the code that is written. So first you basically change your bounds. These are basically changing the bounds. Then you have your model and you set a max value, and then you change your reaction bounds again, and then you change your model objective, and then you try to optimize it again. This is an iterative method. I'll not go into the details, but basically it will give you alternate meaning, like, according to your variability, according to your min and max values, it will give you alternate possibilities of the same objective, in this case, which is aerobic growth of succinate through different pathways. So yeah, this is what FPA will give you. Now, this table on the right is this is the basically 8 variable reactions which are important for this succinate formation. Now, variable reactions for growth on succinate, uptake rate, which is like 20 millimole gram per dW per hour under aerobic conditions. And the minimum and maximum possible flux for every reaction was calculated at the maximum growth rate. So basically, you keep your maximum growth rate constant. Basically, your objective function should give your basically optimal values. So yeah, it's calculated at the maximum growth rate, and only reactions with variable fluxes are shown here. These are different enzymes. They have basically FRD7 and SUCDI. They always have highly variable fluxes in this model because they form a cycle that can carry any flux. Physiologically, these fluxes are not relevant. The other variable reactions are MDH, Me1, which is of importance to us, Me2, NAD transhydrogenase, DPCK, and PYK. PYK is the alternate path that your model has suggested. So these two slides, I just wanted to give you an example of on how FEA is used. So when you add that variability in your solution space, you can get multiple more solutions inside your solution space, which is basically satisfying your objective function or your cost function and this can give you alternate ways to do the, you're basically satisfying the same objective function. So this is what flux variability analysis does, and this is a real life example on which FVA is used. So I just wanted to give you an overview of how FVA works in the real world. So yeah. Okay. Next, we come to the concept of this overexpression algorithm. So we learned about deletions, we learned about dead end metabolites, we learned about blocked reactions, we learned about FEA. Now, like, deletion is a certain type of perturbation. Similar here also, this is overexpression is also a type of perturbation. So uh, like, moving away from the computational side of it, what overexpression does is, it's basically, there are different types of overexpression. You can constitutively overexpress. So basically you turn on a gene and it continuously overexpresses it without any control. So that can be one option. There can be other options, like when you pick a time to overexpress a particular gene, and that is, you're controlling it to the T. So you have a mechanism to overexpress the gene, and after the your work is done, which is resulting of that overexpression, you knock out or knock down that overexpression. So overexpression is generally achieved to see if basically a particular gene uh, is basically performing a particular function. So the general idea is, when you're trying to see if this particular gene has this particular function, so you try to generally overexpress that gene and see if that function increases or not. This is the general concept of overexpression in genes. So overexpression can be done due to different methods, but it will give you an idea about basically the relationship between the gene and the product formation, basically the function, the phenotype. So this is basically what overexpression is. Now, uh, how do you basically incorporate this concept of overexpression into constraint based modeling? That we'll see here. So first, you take the initial fluxes, which is given by this Vj initial, and you calculate, basically you calculate this initial fluxes by constant based flux analysis, which is FBA. You can use using objective function of maximizing biomass mass formation as follows. So this is the maximization function that we all saw in previous slides as well. So you have your V biomass is your, basically, the amount of biomass that is getting formed. You are trying to maximize that V biomass subject to some certain constraints. So you have this constraint: your ATPN should be greater than ATP maintenance, and your ZLYC gene should be equal to your lycopene production. So these are certain constraints that you have put on this. So this example is taken from the lycopene production example only. So there's another. So basically, yeah, it's the same example, but the one that Sir talked about in class. But I'm going into a little bit more details on what are the constraints and kept and so on. So yeah, you have your Sij as a stoichiometric coefficient, basically your matrix for just a second. Sorry. Yeah. Uh, Yeah, your Sij is the stoichiometric coefficient for the metabolite I in the J8 reaction. So it's basically how the structure of your stoichiometric matrix looks. And V initial is the initial calculated flux of the J8 reaction. So now these basically V biomass, V ATPM, V ZLYC, these represent biomass formation rate and the ATP consumption rate for maintenance and the lycopene production rates respectively. These are basically your constraints, or V mass is your objective function you're trying to maximize. These are other constraints that you have put in place for this model. Now, uh, your ATP maintenance is the ATP flux required for a non-growth associated maintenance. And this lycopene is basically V initial lycopene is the lycopene production rate measured from the initial experiment, which is basically you have a number and it was measured by cultivating E. coli in these are details like you can skip. Basically, this is an experimentally derived production rate using E. coli. And it should be noted that in general, this can be provided as a measured constraint or calculated constraint if no experimental result is available. So if you do not have this initial, uh, basically lycopene production rate, you can use it as a constraint. So you can set it a value and use it as a constraint if no experimental result is available. And you basically your V alpha and V beta are basically your upper and lower bounds of the flux of the Jth reaction. So uh, the FBA that we looked at. So you have an upper and lower bound of your solutions, and you have all the solutions inside them. And basically, V alpha and V beta J both are these are lower and upper bounds of the flux of the Jth reaction. And for irreversible reaction, your Vj should go from like basically plus infinity to minus infinity. And for irreversible reaction, it should go from 0 to plus infinity. So it cannot go from to negative because it is basically irreversible. Similar, simple concept. Now, how do you incorporate this concept of overexpression inside it? So this is known as what is known as the FSEOF framework. So this basically schematic sir has also shown. It's from the same paper. So this framework is used to select gene amplification targets for enhanced product formation. So what it does it does is it searches for the candidate fluxes to be amplified through scanning for those fluxes that increase with enforced objective product formation flux under the objective function for maximizing biomass formation flux. Look here, to hear it sounds very complicated, but the whole idea is it's very simple. The concept that is implemented here, if you increase your particular basically flux, is your product getting increased or not? That is your whole idea. So if your particular increase of a particular flux of a particular metabolite is increasing your product formation, that means that can be an candidate for overexpression. So if you overexpress, your product formation increases. That is what it is trying to find out. So it is incrementing it in small batches and trying to see if your growth rate is increasing or not. That is the whole idea. So FSOEF searches for the candidate fluxes to be amplified through scanning for those fluxes that increase with enforced objective. So your enforced objective here is product formation. You're trying to increase your product formation, and you're trying to scan through the candidate fluxes and see whose fluxes are increasing with increasing product formation. It's a similar concept to that. Now, during this implementation, three types of intracellular flux profiles are typically identified: increased, decreased, or unchanged. Oscillating flux profiles can be found in rare cases. So, like you're increasing your production, your growth rate is increasing, but your basically flux is oscillating. That is very rare, but uh, you can generally get increased, decreased, or unchanged. So uh, the increased fluxes, the metabolites with associated with increased fluxes when production rate goes up, those are the metabolites of interest, because if you try to overexpress them, your product formation goes up. So that is what is written here. Among them, FSCOF identifies the fluxes showing the increased profile as the primary amplification targets. Okay, so yeah, this is an overview on how this amplification works. So the overexpression algorithm involves first info. This is an example which is similar to lycopene. This is a different product formation known as taxadine. So it's a similar concept, similar to lycopene formation model that Sir has described. I just took a different example. So if you're trying to find this overexpression targets inside this taxidine production metabolic network. So what you do is first impose a taxidine production flux, which basically should be determined experimentally, but if you do not have, you can put them as constraints. Then you solve an FBA problem and impose an amplification in individual reaction fluxes to simulate the effect of gene overexpression. That is what we saw here. So you try to you search for the candidate fluxes to be amplified through scanning for those fluxes that increase with enforced objective, as in increase with increase in production. And then you solve what is known as, we discussed this last class, minimization of metabolic adjustment problem, and identifying overexpressions that led to a phenotype fraction value, FPH, basically it's given by this parameter, which is the product of the weighted and dimensionless biomass and taxonomic flux values which are greater than 1. Now, these steps 3 and 4 are iterated every reaction within the network. So you this is basically an hit and miss method, right? You think this candidate might be good for overexpression. Now you check if the candidate flux is increasing with the increase in production. If yes, then it is good. If not, then it is. Now you need to do this iteratively to find out which are the best possible candidates for this overexpression. Now for this you have to iterate through these 2 steps. You impose amplification, then you solve for MOMA, and then you find the expressions that are increasing with increasing fields. So yeah, and you make calculations under these assumptions, under the conditions, to simulate complex medium. Uh, these I am not going very detailed into. Basically, these are other details of the model. So yeah, these are other details of the model. So medium composition can be approximated by setting uptake rates of specific chemical components known to exist in the medium of interest. The computational complex problem contained in all 20 natural occurring amino acids, which are L isomers. Uh, the lower bounds of amino acid transport reactions, they took a lower bound. The negative sign indicates metabolite uptake into the cell, and these were chosen upon based on previous literature values, and they were satisfied. They satisfied the relative biomass differences experimentally observed in the media, and also there are some other rates associated with the model. I'm not going into the detail, but this is an overall overview of how you can find for these. You can search for these overexpression candidates. So yeah, this is basically the pathway that is followed, the pipeline that is followed for taxonomic production flux. So you have a base model, you'll add those heterologous components as variability and everything, and then you have fluxes with SPA, and then you add certain constraints and amplify a certain reaction and see if your fluxes are increasing or not. If yes, you identify overproducing strains manually using that particular overexpression. If not, you iterate with a different overexpression candidate, and you see if it is basically giving you a better flux. So yeah, this is how the pipeline looks like. Next, we come to the concept of yeah. Next, we come to the concept of synthetic lethality. So this concept was first proposed in 1922 by American geneticist Calvin Bridges. What it is basically, if you do simultaneous inhibition of 2 genes, basically you have gene A and gene B. When you mutate both of them together, or you knock out or knock down both of them together, that results in cellular lethality. Basically, the cell won't survive. So this is what is synthetic lethality means actually. It is observed when simultaneous inhibition of 2 genes result in cellular lethality. The meaning of the word synthetic, which is used here, is it's not the synthetic artificial synthetic that we think we generally associate with, but it's basically what it means is it's a Greek word which means the combination of entities to form something new. Uh, the combination of entities here are genes, different genes, and forming something new is basically, you can like, is a misnomer in this case because it's not forming anything new, it's just killing the cell, but it's a new cell state, you can argue. Now classical synthetic lethality occurs when inhibition of 2 genes is lethal, while inhibition of either gene alone is not. This is a very important thing to remember. So if you're deleting one gene, the cell is working fine, not fine, maybe there are some disturbances to the cell, but it is not basically dying. But if you delete both the genes together, it is dying. When you are deleting individually, it is not. uh, it, this kind of concept, or this kind of basically idea, can be harnessed to treat cancer selectively by identifying inactive genes, and you target their SL partners. In accordance to this concept, synthetic lethality can also be applied to genes and small molecules used to elucidate the mode of action of drugs. So basically, mm, you 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 try to in case of cancer, you selectively identify those inactive genes, uh, which are basically, you have, say for example, you have a p53 gene which is mutated and is contributed to it's contributing to the cancer. Now you can identify its synthetic lethal target, because that gene is involved in cancer, but its synthetic lethal partner is not. Now that is why it is basically inactive in the context of cancer. If you can identify the gene that is synthetic lethal with p53 and you knock both of them out, it is very possible that the cancer cells will die. You have to do selective knockout in the cancer cells, because if you do it across all the cells in the body, all the cells will die. So that is not a good idea. You need to do selective knockouts or selective mutations. So these kind of concepts, these kind of ideas you can get from synthetic lethals. Now cancer is driven by consequences of mutation or amplification of genes that allow cells to escape mechanisms that control proliferation and survival. Hence, inhibitors that target synthetic lethal partners of mutated or amplified genes in cancer cells can cause cancer cell death without affecting the survival of normal cells. So uh, basically, mutated or amplified genes in cancer, so like this cell cycle control genes or other genes, if you find the synthetic lethal partners of those, you can knock both of them out, thus killing the cell altogether. But you need to selectively do this in cancer cells, so you need to find a mechanism to do that. Because if you do it across all the cells, the whole all the cells will die. So no point in doing that. This figure on the right basically shows a schematic of how synthetic lethals work. So this is an example of cancer. So the principle of synthetic lethality in cancer: loss or inhibition of either of the protein products, gene A or B alone, or overexpression of gene A is viable. So, sorry, my light is not working properly. Okay. So yeah, loss or inhibition of either of the protein products of gene A or gene B alone, or overexpression of gene A is viable. However, pharmacological intervention by the partner gene product will result in SL interaction in tumor cells with a loss of function mutation in the tumor suppressor gene. So basically, your what you're trying to do is you're intervening and you're blocking out this gene A. So your oncogene, sorry, Uh, basically, in your normal cell, you have both gene A and gene B. In your tumor suppressor mutant, a single mutation, your gene B is mutated, and your basically oncogene mutation, your gene B is acting as the oncogene, whereas gene A is in the uppercase, gene B is acting as the tumor suppressor gene. So those are like kind of opposites of each other. So pharmacological intervention by partner gene product will result in a synthetic dosage lethality interaction in tumor cells with a gain of function mutation or overexpression of the oncogene. And mm, basically, the concept I explained. So you have a tumor suppressor gene which is mutated. Now you want to kill off the cancer cell. You find its synthetic lethal partner and you knock both of them out to kill the cell. So whole idea of synthetic lethality. Now, is the most important question comes when talking about synthetic lethals is, how do you identify? You need to identify these lethal targets first. You need to identify the pairs that are together working together can be classified as synthetic lethals. Then you can try to knock them out. First, identification is a major bottleneck for this. So hypothesis-driven approaches for identifying synthetic lethal interactions leverage known alterations in molecular pathways in cancer, targeting specific genes or pathways based on established knowledge. So you kind of do some sort of network analysis or some sort of signal pathway analysis, and you come up with a hypothesis. Maybe these two particular genes interacting together. This is a synthetic lethal interaction. If I knock both of them out, the cell can die. That is a more of a hypothesis driven statistical approach using previous data or previous knowledge. Now you can have experimental approaches as well. So you can use screening based approaches, comparing drug or RNA interference. I hope you know what RNA interference is. Just to give you an overview, so if you have a gene which you want to knock out, or say knock down, you want to reduce the expression of a particular gene. So what you do is known as RNA interference. So there are various forms of RNA interference. Generally, we do currently the most popular way to do is using microRNA. So what happens is, you create a small construct of a microRNA, which is basically reverse complement to the original mRNA you're trying to suppress, and you ectopically express that inside that particular cell or tissues. And what happens is that that microRNA goes and binds to that mRNA, and it does not allow that mRNA to basically convert itself, basically uh, allow translation for that mRNA. It does not allow the mRNA to be converted into protein. So if you have a reverse complement small RNA that is getting stuck to your mRNA, your mRNA will not be basically translated. And not only this microRNA goes and binds, and after that it cuts down the mRNA, chops down the mRNA, and then after chopping down, it moves on to the next target, just like an enzyme. So it's an enzymatic reaction where you have a small microRNA which can basically silence, say, thousands of copies of a particular mRNA you are trying to silence. So this is what is known as RNA interface. There can be other methods as well. So for example, you can have an antisense RNA method. So you have the whole, the sequence of the mRNA you're trying to suppress. You just do a reverse complement of that mRNA in using a plasmid or phage vector. You put that inside the cell. What will happen is, inside the cell, the reverse complement mRNA will go and bind to your target mRNA, but that is stoichiometric in nature. So if you want to silence 1,000 copies of the mRNA inside your cell, you need to have 1,000 such antisense mRNAs that will go and bind. If you have 500, it will only suppress 500. The rest 500 will still be expressed. So there are various types of this RNA interference, but the whole idea is you do not allow your mRNA to function. This is what RNA interference is. So, screening based approaches involve comparing drug or RNA interference treatments in matched cell lines with and without specific gene alterations, allowing for high throughput identification of synthetic lethal interactions, either through chemical library screens or you do genome wide interference screens. So, this is one such approach that is explained here. So there are multiple approaches to generate matched cell lines if a loss of function A or A to C, or a gain of function B to F. So this is basically loss of function. This is basically gain of function uh, of a specified gene study for studying a synthetic lethal interactions in cancer cells that have lost expression of a gene. So here in the cancer cell, you have this X double mutant, which is basically this gene has lost its activity in the context of cancer. Now, the cDNA for the gene can be re-expressed in a different cell line. So the complementary cDNA, you overexpress that in basically another different cell line, and these multiple cancer cell lines uh, both expressing and deficient in this gene can now be investigated. So you have you in your original cancer, your X gene is double mutated, your loss of activity. What you do is you overexpress that gene X cDNA in a particular cancer cell line, and you have another cell line, your original cancer, which is basically X double mutated. So you have two cell lines, cancer 1 having this mutant, and cancer 2 having basically the wild type. The cancer cell lines that express the gene can be treated to inactivate the gene for uh, uh, for studying synthetic lethal interactions in cancer cells with oncogenic mutants that increase the activity of the gene or create new gene fusion. Multiple approaches can be used. So basically what happens is, after you have two types of cells, you can use RNA interference or other methods basically to convert this wild type to a knockout. So and there can be multiple approaches to doing that. And for studying synthetic lethal interactions in cancer cells with oncogenic mutations that increase the activity of a cell or create a new gene fusion, this BCR-ABL is a very popular gene fusion product inside the cell in cancer cells. So there can be multiple approaches to do so. So cells can be treated with RNA interference or a chemical inhibitor to reduce the expression back to or below normal levels. Sorry, guys, my light is just a second. Yeah, it's working now. Sorry, guys. Yeah. Uh, the cells can be treated with RNA interference techniques or chemical inhibitors in order to reduce the expression back to or below normal levels. And multiple cell lines, cancer cell lines, both with and without the mutation, could be investigated. So you have two type. You have a cancer cell line with this mutant, you have a cancer cell line without this mutant. Now you check for which genes are associated with it to find those synthetic lethals. Yeah. Uh, the activating mutation can be introduced or the gene can be overexpressed within the cancer cell line that has a normal expression of a white gene. So basically, if you have a particular gene, you can try to overexpress that. If you have a certain gene, you can try to inhibit that. Using these several combinations, what you are trying to find is what is the synthetic lethal partner of this particular X factor here or this particular Y factor here. This is your uh, target. So if you're trying to mutate one and the other is surviving, the other is uh, all right and the cell is surviving. If you mutate both, the cell should die. That is the idea of synthetic lethals. Now, traditional studies focus on perturbing or deleting single genes or gene pairs, potentially missing higher order interactions and redundancies in metabolic networks. Extending this lethality analysis to triples, quadruples, etc., will capture multiple gene reaction interdependencies, but also faces combinatorial complexity challenges. So it is very evident that like the synthetic lethals, they do not only reside in pairs, they can reside in triplets or quadruples, etc. But when you're trying to account for these, like more than two interactions, like three or four interactions, it becomes computationally and mathematically very complex. So you need to find different combinations. This number of combinations within a vast metabolic network will be very high. So combinatorial complexity challenges will be there if you're trying to extend this lethality, synthetic lethality from 2 to 3 to 4, or if you're trying to increase the number. So this is one paper in 2008, which conducted in silico multiple knockout investigations in yeast and cataloged gene sets providing mutual functional backup of up to 8 interacting genes. So they did up to 8. So that's a very big number. So finding two, it's very hard, but they found 8 interacting genes which can act as synthetic lethals inside yeast. Same author, different year, did a work on basically computation, basically developed a computational approach using game theory for multiple knockout analysis in Saccharomyces cerevisiae to understand metabolic function localization. Another paper here, study here, introduced a framework based on elementary flux models for analyzing structural robustness in metabolic networks applied to E. coli and human hepatocytes. And yeast, with its short non-coding regions and genome containing less than 7% introns, is preferred for genetic interaction analysis, with most research focusing on S. cerevisiae and E. coli. Basically, most research focusing on S. cerevisiae, which is the yeast. E. coli studies are limited to pairwise interactions only. So, and this is a schematic showing a workflow and model-guided identification of compounds that inhibit synthetic lethal protein pairs. So you basically build a metabolic model for multiple pathogens, and you compute this SL pairs for each model and overlapping SL pairs shared among this model. So, and sorry, okay, yeah. So. Yeah, you use some sort of mathematical modeling or metabolic modeling, FPA, whatever you use. You compute these SL pairs for each model and overlapping SL pairs shared among all the models, and then you construct and test double mutants to basically validate your synthetic data. So synthetic lethals meaning if you mutate both of them. So if I have a double mutant, uh, you should not get any growth. And then you perform virtual screening against all validated pairs, and then you test compounds as single agents in the whole assay. So this is the whole overview of like how you can find these synthetic lethals and try to apply them in your research, and trying both experimental and computational methods to knock them out and to study them, how those interactions occur and how they are like basically contributing uh, to the basically lethal phenotype inside the cell. Yeah. Uh, this brings us to the end of the lectures. Basically, the lecture part of the live session. Now we'll solve some MCQs. It's the same method. I'll put it up on the screen. I'll give you 2 minutes, and uh, please write the answer on the chat, or if you want to unmute yourself and give the answer, that is also fine. Because I think only two people are here, right? Yeah. So let's see. Do you have any answers? Both the genes are essential. Yeah, it's true. Because gene A and gene B, this Boolean logic of AND, meaning you need both for this reaction to occur. So if this is contributing to survival, then you need both the genes. Yeah, that's correct. Moving on. Okay, let's see. So gene A or gene B. So you need either A or B, and you need C for the reaction. So gene C will be essential because there's and any one pairs of A at C should be functional. A, C, and B, C. Any one of them should be functional. So option C and B are correct. I think you guys were correct here. Yeah, option C will also be correct. Okay. Okay. The answer is true because the concept of isozyme was different proteins or different enzymes coded by different genes performing the same reaction. So if you have say five isozymes for a particular reaction of an enzyme, now if you knock out one of the isozymes, the other isozyme will take over. But if you delete all the isozyme of an essential reaction, there will be no enzymes to catalyze the reaction. So it will basically kill the organism, which is true.
So, excuse me, sir.
Yeah,
Actually, I have a question on that question.
yeah.
The
Please
previous
say.
question, I have a query on your question, sir.
Sorry, I can't hear you properly. Can you just speak?
Can you hear me, sir?
Yeah,
Actually, I have a small query on the previous question.
yeah, yeah.
So, like, they have mentioned if you are deleting an isozymes of an essential reaction,
Hmm,
it may kill the enzyme. So, what I will think is like isozymes are like the enzyme which is having another form similar
hmm.
to that, right?
Yeah, yeah,
So, like, if only the isozyme is being deleted means, then there are some other enzymes which are similar to that, so it can
yeah,
survive, right, sir?
yeah, yeah. But the question here is basically different. The concept you're thinking is perfectly all right because isozymes are different forms of the same enzyme. So if you have five isozymes for a particular reaction, if you delete one of them, either of one of the other four will take over the reaction and will catalyze the reaction. But the question here is the deletion of the isozymes of an essential reaction. So basically, you're deleting all the isozymes of an essential reaction. So if you delete all the isozymes, so there is no enzyme to catalyze that reaction basically. And if it is an essential reaction, the cell will not survive if there is no enzyme to catalyze. So your concept is correct. If you delete one isozyme of the family of 5 or 6 or 10 isozymes, then the other isozymes will take over. But if you delete all, there is no enzyme to catalyze that reaction. Do you get
Okay,
it?
here the case is like we are deleting every
Ah,
isozyme
yeah,
specific.
yeah, yeah, yeah. The question is not very clear, but what it is trying to say, you're deleting
Okay.
all the isozymes of an essential reaction.
Yes, sure. Thank you, sir.
Okay.
Yes.
Yeah, it's true. So your R9 is basically converting your M8 to M9 plus N10, and you need both gene A and B. And if your R9 reaction does not carry any flux, as the metabolite M8 is not produced when glucose is used as a substrate. So basically, your M8 is your basically M8 is a basically dead end metabolite when glucose is used, and when xylose is used, the R9 will carry flux. So basically, M8 is a dead end metabolite when it is glucose, and M8 is not a dead end metabolite when it is xylose. R9 will carry flux. So this R9 is basically conditionally blocked. So when you are using glucose, it is a blocked reaction. When you are using xylose, it is not. So that is what a conditionally blocked reaction means. So yeah, R9 will be a conditionally blocked reaction. So that's true. Yeah. Okay. 9 is dead in metabolite. Yeah. So there is no reaction that is getting basically producing uh, M9. Uh, and there is no reaction that is consuming M9 as well. So it is a dead metabolite. M8 isn't, because there's consumption of M8. Production of M8, there is no like what to say information on that, but consumption of M8 is there. So M8 is not a dead metabolite. M9 is. So yeah.
But like actually, my thought process is the same, but at the same time, like M9 is also being consumed, right, in a way. But since it's not getting produced, it will be dead end. But I was sort of thinking, like, if this is the whole system, wouldn't M8 also be a dead end metabolite?
Yeah,
If this is the whole
if
system,
you think like that, yeah, because you see there is no reaction here producing M8, and
yeah.
like
But at the same time, M8 and Mn both are getting consumed in a way.
M8 and M9 are both are getting consumed, and M9 is not getting or what to say
Hmm.
like M8 and M9 are both not getting produced. So if you think of it like
Yeah.
that as a closed system, yeah, both of them are dead end metabolites. But here I am not I am also not very sure about the answer they have
Hmm.
provided. But yeah, the concept is either it shouldn't get produced or it shouldn't get used. It's a dead end
Yeah.
metabolite. That's the concept. But
Yeah.
yeah, if you think of this as a closed reaction, then both M8 and M9 should be dead end metabolites.
Yeah.
Yeah, that's true. So, like, an iffy question. Sorry for this. Sorry. Yeah. Yeah. This is a very straightforward one. The answer is no because false or no whatever. It's because your R5 is bound between 0.23 and 0.89. So you have a minimum flux and a maximum flux associated with your FEA. And under the same conditions, it is not possible for the reaction of the flux to exceed your maximum flux. Your 0.97 is more than 0.89. So it's not possible to exceed under the same conditions of your maximum flux. So yeah, it's not possible. I'm sorry. Uh, um, R3, R4, R12. I think it is correct. Okay, so just to give this to like go through it. So you have M1, M2, M5 in your growth medium, and basically M1 here, M2 here, and M5 here. M3, M4, and M9 are getting basically produced here. Now R3 and R4, so these two, these are basically producing M9. So which basically M9 is what is getting the biomass. M9 is basically contributing to the biomass, and so sorry, uh, yeah. So so R3 and R4, these two reactions are producing M9, which is basically contributing to biomass. So if you delete uh, um, R3 and R4 together, these are basically double lethals. Now, R1, if you check, R1 is producing M3. If M3 is not produced by R1, there is no other source for M3 in this reaction. So if M3 is not produced, M4 will not be produced, and M9 will also not be produced. So R1 is a single lethal. Uh, I think there were one more
I
option.
actually added one more option like R2 and R5
Okay,
because
let's check.
so my thought process was in
R2,
R2 is the only
R5.
source by which M4 is created.
Hmm. Uh,
But my
but
like the confusion
R2
for
will
me,
be then a
yeah,
single lethal, right?
yeah. But then my question, like, but because M4 is like M3 is required for M4 protection and M9 protection. So R1 is definitely a single lethal. But without M4 also there is this R4 reaction where M9 is getting produced just with
Ha,
M3 and
but
M9.
this M4, the M4 reaction, this is a single lethal reaction. Na? So if you say, for example, stop the R2 reaction, M4 will not be formed. So it's a
But
single reaction.
it
But
doesn't stop growth in a way, because if M4 is not there, there is an R4 reaction where M3 plus M5 again produces Ma. But for me, the confusing part was this like R5 reaction where I was wondering if R5 is not there, so will it stop growth essentially? Like
no, R5, the R5 reaction is primarily it's not a, if you look at it that way, it's not a reaction per se, because
yeah, yeah,
your M9 is getting produced, which is, if you consider your flux balance analysis, you try to maximize your
mm,
M9. That is what you're trying to
mm.
do. The M9 to biomass
Yeah,
conversion, that's not a actual reaction as per se, because
yeah.
you're concerned with the production of M9, not the biomass.
M9, yeah. No,
Yeah.
because it was explicitly written like there's this R5 reaction
Yeah,
as
yeah,
well, so
yeah,
I
yeah.
started getting confused. So
Yeah,
I
but
was sort of
it's not a reaction per se, right? If M9 is getting
yeah, yeah,
converted
yeah.
to, you'll ultimately look to maximize your M9 only, right?
M9 production, yeah.
So that will be like maximizing your R4, not R5.
Yeah, yeah, yeah.
Okay. Yeah. So yeah, R2 can be like called a synthetic lethal, but again, as you said, like M4 is required for M9, but M9 is also getting from M3 and M5. So
Yeah, yeah,
yeah.
yeah, yeah.
Okay. Why not Gene A? Gene A is also a single lethal, because if you delete Gene
Yeah.
A, so your M3 and
There's no
M4,
M3, M4, yeah.
yeah. So
Yeah,
yeah, the other options are correct. So if you delete Gene F, there is no formation of M6, which is your like
yeah.
objective. Gene A also, if you delete, you won't get M3 and M4, because
Or
these are the
M4,
only
yeah.
sources. Now C and E, if you delete, basically you're deleting this reaction, so you're converting M3 to M5 and M7, and ultimately M5 is getting
But
converted to
M5 is, yeah,
M5. You can form
but
from this
M5 is
reaction,
getting produced. Yeah,
yeah, third
yeah,
reaction. So this is not a double lethal.
yeah,
And if you delete C,
yeah.
D, E, so you are deleting
Then
this reaction and this reaction. So M5 will not get formed.
yeah,
So yeah, yeah. So
okay,
ultimately M6, no formation of M6.
yeah,
Okay. Okay. The answer is correct. It will be reaction one. So the general concept of basically FSEOF is you try to increase the amount of biomass, or the basically your objective function, what you're trying to maximize. You try to maximize, try to increase that metabolite, and check your model and see which flux is increasing basically. So you have your fraction of the product on x-axis, so your point 0.1, point 0.2, point 0.3, and point 0.4, your fraction of products is increasing, and on the y-axis your given is your flux value. So if you look carefully for reaction 1, at point 0.1 fraction of the product it is low, then it increases to point, it increases at point 0.2, it increases further at point 0.3, and further at point 0.4. So with increase in fraction of the product, reaction 1 flux is increasing. That is not the case for other reactions. So for example, reaction 2, it is kind of constant across all the across increasing fractions of the product. Is an a kind of an opposite reaction to what is happening in reaction 1 is reaction 3. So it is kind of decreasing with increasing fractions of the product. And reaction flow has an haphazard trend. There is no trend actually in reaction flow. So yeah, with the only reaction that is increasing flux with increasing fraction of product is your reaction 1. So yeah, uh, reaction 1 will be your possible target for your overall expression. So this brings us into today's live session. I'm sorry for the noises. This brings us today to the end of the live session. If you have any questions or anything, you can ask, or we'll close this session.
This is Vishal.
Yeah.
So actually, like, is it possible to conduct some software kind of sessions to hand out and like overall view of like go through something like basics?
So, like you are talking about Cobra or any other software?
So like until now we haven't came across like any lab kind of system.
So you have a lab session in for Cobra, like for this week also, and next week's also you have, I think, a couple of lab sessions for Cobra. So Cobra is a toolbox from Python. So it's Cobra Pi that is used for this FBA, different types of this basically flux analysis and creation of this model. These type of things are done using that Cobra toolbox. So there are inbuilt models. You do not need to even like kind of build your own model, build your own objective functions. You do not need to do that. Under different situations, different models have already been created, which you can use, you can use inside the toolbox, and there are various options. So this, just a second, I'll share my screen again. So, yeah, basically this example that I showed for FVA. So here you're getting two alternate pathways. One is through ME1, one is through PYK. This is how your FBA works in Cobra Toolbox. So your Cobra Toolbox will have these functions. So you can change your reaction bounds. You can change your models. There are like inbuilt models within FBA in basically Cobra Toolbox. You can like the very simple steps because everything there is pre-built. So and I think there are lab sessions for Cobra Toolbox for this week as well as next week. So I think you'll get enough exposure on how to use Cobra Toolbox. I can like what I can do is I can have a like a little bit of explanation of how those things work, but I'll have to take that on upon the like last class of this basically course. So here you have 12 weeks, so for each week I have 2 hours to basically have a live session. So total is 24 hours, and at the end of the course you have 1 hour for like covering it's basically giving you an overview of the whole course and covering topics from where you have doubts and stuff. So I can cover the Cobra Toolbox in that session if you want, but I think you'll get enough hands-on from the lab sessions only inside the course. But yeah, if you want, I can have that then, because before then I would think don't think I'll be having much of a time to handle that. So if you want, I can have that then. So you want to learn for the Cobra Toolbox only, right? Or any other software you have in mind?
Yeah, as you mentioned that in last after completing the 12 sessions, we can have like go through every software kind of session. That would be better, right?
That we can do at the end of the course. Like
Yes.
after the 12 weeks are completed, there will be one session where I'll cover basics of the entire course. So in that session, I can give you a more uh, a better overview of like basically Cobra Toolbox and other software. MATLAB, I am not very confident in MATLAB, so I'm not
Yes.
sure if I'm the ideal person to go through MATLAB softwares. But Python I can handle. Cobra Toolbox I can help you with.
Sure. Thank you.
Okay, okay, okay then. Thank you. I'll close this session now.
Thank you.
Yeah, thank you. See you next week.